Right, we're in this second part of chapter 19, which is on genetic technologies. And so today we're going to look a little bit more at applications of transgenic organisms and look at different methods for inserting DNA into an organism. This is kind of an extra little bit I'm adding into this chapter. So that was one of your questions on your study guide, and some of you probably didn't know how to answer it just because it wasn't really in this chapter. So I'll be giving you your worksheets back so you can kind of finish that on Tuesday when we get back together again. DNA microarrays and what they're used for, and then the difference between gene silencing and genome editing when we try to manipulate the DNA. So we already talked earlier that a transgenic organism is one that has DNA from another organism inside it. And so we can use transgenic organisms in a number of ways. And one of the ways is what's called farming. And you notice the spelling here with the PH. So that's dealing more with pharmaceuticals. And so it's a way to have organisms change them by inserting a gene from another organism that allows them to express some sort of recombinant genes in a body fluid of a transgenic organism. So oftentimes this will be different genes, oftentimes human genes, that can be expressed in the milk of the animal, such as transgenic sheep, cows, and goats. And so things like clotting factors, clot busters, collagen, various antibodies, we can have that secreted in the milk of that transgenic organism. So it's a good application of a transgenic organism. Now, how do we get the DNA into that transgenic organism? These are like the 5 main ways here that you should know about. The first one is just using chemicals. What chemicals do is they open up transient holes. That just means temporary holes. So the chemical allows the plasma membrane to just the pores to kind of open up temporarily, allowing that DNA from the other organism to be inserted into the new organism, and then it seals back up again. Another one is liposomes. If you think of liposuction, we're talking about fat. So liposomes help carry the DNA into the cells on fat bubbles. Okay, remember your phospholipid bilayer, those phospholipids are made up of that fat, or the lipid portion of it, so it allows those to transport through a little bit easier and carry the new DNA in there. Electroporation, like the name kind of suggests, it uses an electric current to again open holes temporarily in the plasma membrane, allowing that DNA to enter. Particle bombardment, or what sometimes is referred to as a gene gun, basically they're teeny tiny metal balls that are coated with the DNA, and then they basically get shot into the cell. I've seen this done up at NDSU when I was doing my research for my graduate degree in the genetics, barley genetics lab. And it's shot so quickly inside this little encasement that you can actually see they were trying to shoot the DNA into the barley, beginning stages of a barley plant growing, and you can actually see the leaves in the plant just kind of jump almost as those little BBs, so to speak, are being shot into there. Now you think that would destroy the cell. It does maybe for some of them, but most of the little tiny metal balls do get in. Again, it happens so quickly, it goes into the cell and then, basically making a little hole, but that seals up right away of the plasma membrane. And so the DNA gets put in that way. And then microinjection is the use of a small microscopic needle to basically inject the DNA into the cell. So all of these kind of tell you how the DNA gets in, but you should know these 5 ways of inserting DNA into a new cell. So once you've altered that cell and added the new DNA, the organism must be regenerated from that altered cell. Now, a couple of things happen here. If the trait the gene you're trying to get in, if that trait is dominant, the transgenic animal must express it in the appropriate tissue at the right time in development. Okay? If the trait is recessive, then they often have to do a cross between two heterozygotes to yield the homozygote that can then express the trait. So even though we get it into the organism, we might not be quite done yet, depending on what gene it is that you're inserting. Why would we ever use transgenic animals? Well, sometimes they can be useful as models for human diseases. For example, inserting the mutant human beta globin gene that causes sickle cell anemia into mice, and then being able to test various drug candidates on the animals before they have to test it on humans. And if they see any significant side effects, then they can just abandon that and say, nope, we're not even going to try that on humans. Now, there are some individuals who believe that's cruel to test on animals as well. And again, I think they're just trying to find ways to safely, before they ever bring it to humans, see if they can test it. So even when they're testing on animals in the lab, they do have to follow strict protocol as far as trying to do it as humanely as possible. There are some limitations with transgenic animal models. Sometimes they don't have any control where the transgene inserts or how many copies do so. So that might cause issues down the road. The level of gene expression necessary for a phenotype may differ in the model versus what humans need, and the animal model may not mimic the human condition exactly because of how we develop differently. So while transgenic animals may be useful to some extent, they're not perfect, and they're not exactly like humans, so it's not 100% effective. Other things that sometimes you might hear about are genetically modified foods. Usually when food is going to be genetically modified, it's usually based on phenotypes such as taste or appearance. Genetically modified organisms, when we talk about GMOs, they have altered genes from other species to over or under express their own genes. Again, genetically modifying foods, which are usually in form of plants and so on, usually field tests may not adequately predict the effects on an ecosystem, so they're not sure if we suddenly introduce a transgenic plant into a field or an environment, could some of their pollen or other things spread to other organisms, especially people who are trying to keep their fields or their crops organic and not using other things. So that's always kind of a fear. Also, the idea of genetic uniformity. The nice thing about nature and plants growing in nature is we have variety, and as different organisms cross-pollinate with other ones, we get some of these various different variations, and that's what natural selection and evolution is all about. If we start to use transgenic organisms where everything's the same, and pretty soon we don't have that variety, that could be a little bit scary too. Transgenic organisms can also provide processes as well as products, and so one term that we use here is what's called bioremediation. I think we had this way at the beginning at chapter 1. It's the use of bacteria or plants to detoxify environmental pollutants, such as cleaning up soils that have various contaminants in it, nickel, mercury, the TNT in landmines. So we can make transgenic organisms to help try to clean up those areas. How we can actually monitor whether or not the gene is functioning as it should be, we can use things called microarrays or gene chips. They can detect and display the messenger RNAs in a cell. And then that messenger RNA, however much is expressed, can kind of, remember, that's going to be used to be translated into the protein then. And so they just use these little pieces of glass or plastic. It almost looks like a little, what do they call it, cover slip, like on a microscope slide. And many small pieces of DNA can be attached to it of a known sequence. And then they can take a look at it and they can compare a sample from an abnormal situation to a normal control to see what's happening there. So messenger RNAs are extracted from your sample, and then they make copy DNAs, and then they can use different fluorescent kind of tags or dyes on them to label them, and then they could apply it to the microarray and they can look at the pattern and color to see which genes are expressed. And then they use a laser scanner and computer algorithms and all of that to try to interpret the results. And so it kind of, first of all, they take a control sample, they compare it with somebody who has maybe an injury where they can isolate the RNA. Here they're doing a spinal cord injury. And then they use reverse transcriptase to generate copy DNA. And then they label them with those fluorescent tags. And then they incubate them on that little slide. And then they use a laser scanner to look at the different fluorescent probes, and they can analyze, based on all the color arrangement, what's happening there. So they can look at, let's say, somebody who's had a spinal cord injury, they can look at gene expression of the various RNAs to see if there's any repair going on in that spinal cord, in those connective tissues and so on. And so they can kind of analyze that. Last little bit here is on gene silencing and genome editing, which is very kind of our new recent development. Gene silencing, just like the name suggests, blocks the synthesis of or degrades the messenger RNA. Again, if you destroy or block the messenger RNA, you're not going to be able to have translation, therefore no protein can be made. And so you have, in effect, silenced that gene. Genome editing allows you to create little double-stranded breaks in the DNA double helix, and then you can insert a desired DNA sequence or remove a particular sequence that you don't want in there. And so genome editing kind of opens up this whole new realm of, could we someday, in a sense, treat and cure people who have genetic disorders where, you know, the sickle cell anemia, the cystic fibrosis, could we go in, remove that abnormal gene and replace it with the normal gene? That's kind of the future of genome editing. That it's pretty exciting, and probably in your lifetime these things might be found. So the three that fall under genome editing are right here: the zinc finger nucleases, TALEN, and CRISPR. And then the gene silencing, where they block translation, are the antisense oligonucleotides, ribozymes, and RNA interference. So genome editing, just briefly, I'm not, this is so new. Haven't even had a lot of background knowledge of this, but the zinc finger nuclease technology, they use parts of proteins called zinc fingers. That's why it's called that. They bind to 3-base DNA sequence, and then they cut the DNA so genes can be swapped in or out. Okay, and then TALEN does pretty much the same thing, but uses a specific restriction enzyme from a bacterium. All of these, their goal is to cut out a certain section of DNA and maybe swap it out, remove the bad gene, or swap in the good gene. What's being used more often, I think, is what's called CRISPR technology. There's an enzyme called Cas9, and it actually uses RNA that's complementary to the DNA strand, and then it can cut out that DNA and insert it with the appropriate strand. And so, exciting stuff. Like I said, hopefully in your lifetime they'll be able to maybe really treat and help people who have some of these genetic disorders, that especially the ones that are only like one gene, basically one base that's altered. Okay, RNA interference is where we use RNA molecules, just like the name suggests, they interfere with translation, basically. So there are RNA molecules that can fold into short double-stranded regions, just like it shows right here. And then those short double-stranded RNAs are sent into the cells, and they separate into single strands. And then one of these strands will bind its complement in mRNA, preventing it from being translated. So that's why we call this RNA interference, because it basically lays down that short little piece of RNA and blocks it. It binds to it, and so it cannot be translated into the protein. We can't, transfer RNA can't come and attach to it and bring the correct amino acids to make that protein. And the small RNAs that do this are called small interfering RNAs, or siRNAs. We also have antisense induced exon skipping. What this does is it silences mutations, okay? So it helps stop the mutation from occurring that causes exons to be cut out of maturing messenger RNA. Remember, it's your exons that code for proteins. So if you have a mutation that actually causes the exons to get cut out before the messenger RNA leaves the nucleus and goes to a ribosome, you're never going to have that protein made. So what researchers have done is they make these synthetic DNA molecules called morpholinos, and they have a complementary or antisense part to the gene that has that splicing mutation. And so they bind to the complement, and then the original mutation is bypassed. So now that protein can be made. In other words, the exon does not get spliced out. It can stay in there, and your protein gets made. And so that's another way of being able to kind of alter the DNA so that your proteins can still be made. And that is it. It's some complex information, but very interesting and exciting for the future, especially for people with genetic disorders.